Hi, I hope everybody can hear me. I think we're going to start now. So let more people will probably be joining in, but we'll begin. So hi everyone, I'm Bhavya from the Biodiversity Lab in NCBS. And as part of Big Butterfly Month, I will be talking to you all about how butterfly wing color patterns evolve. And the title I've picked specifically is because the word kaleidoscope itself is the common noun that is used to describe a group of butterflies. So if you see a group of butterflies flying around, it's called a kaleidoscope. So let's get into the diversity of color patterns that we see in biology, in nature. So just a minute, sorry, my slides, yes, yeah. So we see a lot of diversity in color across the scale of biology, from aquatic organisms to terrestrial, arboreal, all kinds of organisms. We see that these animal colors can be of ranging from the diversity of one end of the spectrum to another. We see blue animals and violet and purple animals and flowers and plants all the way up to red and orange. So an interesting and scientists have been really intrigued by this diversity question for a very long time. And humans are also highly visual creatures, and we really get enthralled by a lot of colorful organisms and are attracted to color in general. So this has definitely been one of the main reasons also why I think that scientists have been trying to understand color in biology. And it's this interest has been there for centuries now, and people and different scientific groups have tried to understand questions about mechanisms of color production. For example, what exactly is causing, like how are these colors generated, and also related to function of these colors, basically what are the functions that different colors perform, and also how these different kinds of color patterns evolve. And since this has been studied for quite some time, you might think that, a lot of these questions are actually already answered. But the thing is that there is still a number of unanswered questions that are present in biology with respect to coloration and color science in general. So let's get into the idea of color science and the study of color in nature. But one of the ways that we try to understand the diversity in color patterns, or basically how we understand the processes that shape diversity in nature, is through understanding the color through its function. So basically, one of the ways that we want to understand what a function of a color, why there are so many color patterns, is through the understanding the function of these color patterns. And what exactly does that mean, right? So in a very general sense, color functions as a visual signal that can convey some amount of information to the perceiver of this particular color. And to get into more biological senses, broadly, we're trying to answer the question, what functions do these colors serve? And the idea is that these color patterns can serve functions ranging from protection to communication and to regulate physiological processes like thermoregulation, etc. And what I'm going to be doing right now is focusing on a few of these aspects at the different parts of my talk, and try to try to put forth the point of how we can understand diversity through function. So let's take a look at protection in the beginning itself. So asking the questions, how do these colors serve the function of protection? One of the ways in which this has been established is through this phenomenon known as camouflage or crypsis. And this is a process in which an organism blends with its background and it remains undetected. So basically, you can't really identify this organism amongst the background that it is present in. And this kind of a phenomenon is used by both prey and predator organisms. And for prey, it's often protective, which is what the idea is, that you go undetected by your predator. But in the case of a predator, they also use camouflage to sort of lay in waiting for their prey and ambush their prey when the prey comes closer. Because again, it's blending with the background, and this can work in both ways. And for example, here we see this Draco that is being very well camouflaged against the bark of this plant, of this tree. And we know that it's also one of the most widespread anti-predator defense systems that we see in all organisms, ranging from aquatic to terrestrial, arboreal, aerial, everything. And I just want to highlight that point by showing you all this video of this octopus that is doing an insane camouflaging tactic here, where from a distance, this just looks like a normal underwater plant that is existing and doesn't look like there's much happening here. But as the diver's going closer, we suddenly see that there's this octopus that's just been lying there and is now sort of evading the diver and swimming away and trying to escape from the diver. So from this, I hope you can see that it's a strategy that is used by a lot of organisms, and an octopus is a great example, really. So moving on from camouflage and crypsis, there are multiple other strategies, and the second one I'm going to be talking about is this strategy called masquerade, where it's the phenomenon when an organism is misclassified as an unprofitable food source. So what do I mean by that? It's that an organism looks like something that is inedible. So it could look like a twig, it could look like a leaf, it could look like bird droppings. And basically, if a predator sees these colors on an organism, it's going to think that it's something unedible and ignore it, and therefore not predate on this particular organism. And giving you examples of how even plants do exhibit these defensive strategies. On the left side of this panel here, we see the plant with these white markings, which makes it appear like a leaf boring insect has actually damaged some of the leaf. And thereby this could deter other herbivores and other insects from laying eggs on these leaves, because it also already appears damaged. And on the right-hand side, we see the presence of this ghost mantis, which looks like a twig. And it could again be misidentified and therefore ignored because it just blends with the background and looks like a twig. And as you all must have already guessed, that this both masquerade and the previous phenomenon of crypsis or camouflage, they can often work together, and these organisms can be undetectable or can also be misclassified in terms of masquerade. Another sort of popular protective coloration that we see in organisms is that there is bright warning coloration that is used to advertise unpalatability or toxicity. And this is a phenomenon known as aposematism. And this is more of an active phenomenon where you have an organism that actually displays very bright colors and advertises that it cannot be eaten. And again, this is widespread across the tree of life. We see frogs, we see snakes, sea slugs, butterflies, ants, a lot of organisms that actually exhibit aposematism and or warning coloration, as it's commonly called. And an outcome of aposematic coloration is another phenomenon or another defensive strategy, which is called mimicry, in which an organism evolves a resemblance to an aposematic species. And this thereby gains protection by looking like a toxic organism. And there are two types which I'm going to be talking about, but there are multiple others. But for the sake of this talk, I'm going to restrict it to two kinds of mimicry. We see one kind of mimicry called Mullerian mimicry, which is an evolved and honest resemblance between two or more aposematic species. Basically, both these mimics are toxic, but they share the same color pattern. Illustrated here through this example of some beetles in Asian netwing beetles. Both these rows are of toxic individuals, and they share the same color pattern. If you look, this is the same yellow as this, and the black and yellow banding patterns are similar here. And we also see this in the case of millipede mimicry rings, where we have the same color pattern that is shared across different species, but both these species are toxic, or all these species in this case are toxic. Another kind of mimicry, which I said I'd be talking about, is Bateson mimicry, which is a deceptive resemblance of a palatable or an edible species with a toxic aposematic species. And this reduces the predation pressure on the palatable species as well. And this is a slightly negative interaction because the model or the organism that is aposematic is not really getting any benefit from the Bateson mimic existing in the environment. And this is exemplified by these examples here of the coral snake mimicry ring, where we have a coral snake that is toxic and this scarlet king snake, which is not toxic and actually edible. Similarly, here we see wasps and hoverflies, of which wasps are considered to be the models here, and hoverflies are the non-toxic Bateson mimics. But so from all of these examples that I've spoken about, I hope that I have sort of been able to highlight that color patterns in this case, in terms of protection, serve as an anti-predator defense that is shaped by natural selection. So by natural selection, it's nothing but this cross-species interaction that I'm referring to with prey and predator, where a predator exerts some amount of selection onto the wings of onto these color patterns in these organisms. But So far, I've been talking about almost everything except butterflies, and this is supposed to be a big butterfly month talk. So what exactly is happening in butterflies as well? It turns out that butterflies also employ these same strategies, and we see that this is just a sort of I was just trying to give you all a general introduction into coloration strategies across which can be used in any kind of organism. We see that camouflage and masquerade also exist in butterflies. For example, in this common evening brown butterfly, which is very prevalent in Bangalore and other nearby areas, we see that it's really well camouflaged with its background of leaf litter. And similarly, we see masquerade as well in not just the adult butterflies, but also in the larval stages, where this orange oak leaf butterfly resembles a very dried up leaf very closely. And this particular instar of this of the common Mormon butterfly, which is again very common in Bangalore and nearby areas, resembles a bird dropping, and therefore any predator is not going to be interested in eating these individuals. But one of the most common and most widely studied phenomena or protection strategies that are seen in butterflies is actually of aposematism and mimicry. This is very widespread in butterflies, and we see a lot of groups of butterflies from South America and even in India, a lot of groups that employ aposematism and mimicry as a defense strategy. And I'm going to just show you four examples of mimicry rings here that we find in the Western Ghats of India itself, where we see, if you look at the first row, we have the aposematic individuals or the Mullerian mimics in this case, where we have two individuals that share a color pattern but are both toxic. And we have, within the black color boxes, we have the Batesian mimics that are copying these color patterns. So we have more mimicry rings that are present in Western Ghats, but I'm just showing you four here. And you can see that there is, again, both aposematism mimicry and Mullerian mimicry and Batesian mimicry over here. But what I am not really talking about is these other sort of butterflies that are present outside of the box. I haven't really mentioned anything about, say, these individuals here. And if you look at this diagram, you can see that I've marked some as females and some as males. So all the individuals in these boxes are actually females, and the individuals outside are males. So what really is happening, right? Why are only the females participating in these mimicry rings? What is up with the male wing patterns? And why is there so much of a difference in some cases between males and females? So that's exactly like one of the questions that a lot of groups have also tried to understand. Like, why are there male female differences in wing patterns in butterflies? But let's just put this at the back of our heads for now. Let's come back to this question. And I want to sort of move over again back into the function of colors and trying to understand the diversity through function. And all this time I've been talking about how color can act as a communication signal between a prey and a predator. So basically two different species. But here with male and female, it's the same species, right? So color patterns can also mediate intraspecific communication. So between males and females of the same species. And this actually is very important because males and females need to be able to identify each other as the same species. And color patterns can often be used as a recognition cue, along with, of course, olfaction or smell and other kind of morphological cues like size and other presence of horns or other kinds of things. But again, this is not just it's not just for species recognition. Another function that has been attributed to color patterns in butterflies and other organisms is that the variation that we see between male and female can be used as an important visual cue to help individuals choose mates. And this is where the idea of sexual dimorphism or colors being used as ornaments comes in. And I'm going to explain to you what sexual dimorphism and these colors as ornaments mean. We have sexual dimorphism, which is nothing but the difference in a trait between the males and females of a species. So if you go back to the slide that I had shown you and you remember those butterflies that I showed you, we have females that participate in the mimicry rings with males having very different wing patterns. And just to sort of have a more common example, the peacock that we all know of, the males are very blue and they have a bright color of feathers behind them, and they are also larger in size. But the peahens are very small and usually brownish or whitish in color. And this is exactly what is meant by sexual dimorphism. There's a difference in size, difference in color, there could be difference in other characteristics as well. And then what I mean by coloration can act as ornaments is nothing but these color patterns can indicate the quality of an individual. And ideally, an ornament is something that is just added to make something look more attractive. And that's exactly what coloration can also do, where the presence of a good looking ornament on an individual can result in that individual getting more number of matings. So it can be used as a signal that indicates the quality of a individual, and can be used to choose appropriate mates subsequently. And sexual dimorphism in butterflies can be through their patterns. That is how these color patches are exactly arranged. Is it a white on a black? Is it a striping pattern? Is it different kinds of patterns that are arranged differently? Or it can be just within a color patch itself, restricted to one particular wing element. And if I go back to the example of the mimicry rings here, we see that in the case of these butterflies that are present in the lower row, we have two different kinds of females that have different color patches, different color patterns on their wings as well, as a result of mimicry arising in them. They have a completely different forewing and hindwing pattern from the male over here. And so here you have dimorphism in both color and pattern, and the appearance of a red, for example, in these females, but the male doesn't have it. So there is color and pattern dimorphism in this case. But we can also see that there is just dimorphism in a single color patch alone. And if you look at the two panels of butterflies that I've shown here, which is a yellow pansy, again, very common in Bangalore, we can see that the males have the presence of this bright blue patch, but the females, while the rest of the color patterns remain the same, this particular blue patch where it's supposed to be has been covered up by black patches. So this is what I mean by there is dimorphism in a color patch and versus a dimorphism in a pattern. So I hope like with this, there is an idea of why there may be male-female differences, mainly because it serves as a communication channel between the sexes of a single species. And I've been talking about dimorphism a lot in the color and pattern, but it's still very limited to just the visual or the Vibgyor range, which is the rainbow that humans are sensitive to, from violet to red. And that's mainly because humans can only see have only three photoreceptors, or we can see basically red, green, blue, and a combination of these colors. We can't see what's beyond these ends in the spectrum of light that actually reaches the Earth's surface. But butterflies and a lot of other organisms also tend to have an interesting additional color, which is a color in the UV. And I've illustrated two examples here of Urema hecabe and a Coleus eurythium butterfly, where we see that under normal visible light, I'm just going to change this to a pointer actually. Under normal visible light, both males and females look exactly the same. This is like you take a normal photograph of these butterflies and they look exactly the same. But if we were to use a filter that sort of cuts out every other wavelength of light and only allows the UV light to pass through, we see that the males and females are actually very different with respect to UV coloration. And their ventral side also is the same. But, and again, over here in this Coleus butterfly, we can see that under normal visible light, that is if you normally take a camera and photograph these butterflies, they look very similar to each other. But under UV color, under UV illumination and allowing only UV light to pass, we see that the males have a very bright UV patch on their dorsal wings, but the females don't have it. So we see that in addition to dimorphism in the visible range of the spectrum, we can also see dimorphism in butterflies in the UV region. So I've spoken to you about colors in the visible range, colors in the UV range. So I'd like to take a small digression here and talk about the types of colors that we actually see in butterfly wings. That basically is what is the UV color doing? What is it made of? And what studies have been carried out to identify colors in butterflies. So in addition to colors in the visible region, which are predominantly pigment based. And what I mean by that is that they have small molecules or small chemicals that are deposited on the scales of butterflies. Butterflies also have UV coloration as illustrated in the previous example. And along with the UV coloration, we also see the presence of strongly iridescent color. And iridescence is all of this metallic sheen that you see in some of these butterflies, illustrated here by this striped blue crow butterfly. And if you look at this butterfly, the blue region here is what we're referring to as iridescent coloration. And often the iridescent coloration that we see, which gives rise to blues and greens in butterflies, is not based on a pigment or any chemical that is being deposited, but is actually dependent on the structure of scales themselves and how scales are arranged one on top of the other, that brings about the reflection of light in these different ways. So there's no pigment that is bringing about this blue in this butterfly, and even in the UV coloration, it's not a pigment that is causing UV light, UV reflectance to come up. And in addition to this, we also have colors that are fluorescent, and what that means is that they are normally, they have one color under visible light, but under certain other illumination, they may appear to be a slightly different color. So for example, here under visible light, this butterfly has a normal black and white patch, but under UV light, we see that this butterfly actually has a fluorescent blue color. So the white actually emits blue color wavelengths, and then that is what we see as fluorescence. And again, this is a pigment based coloration, and our lab is actually has worked on trying to identify some of this chemistry behind the pigments that are causing fluorescence to also occur in butterflies. So now I've sort of sidetracked and taken you all into the types of wing colors that we see on butterflies, but let's head back into the dimorphism range and the functions of colors again. So hopefully from the dimorphism that we've spoken about and the functions between that, I hope to have motivated the idea that these colors have been found to be used as mating signals, where individuals with maybe brighter colors or where the patches are bigger or larger will obtain more number of matings. And from this, I think we have established that there is a potential second function to these color patches, where they serve as intraspecific communication signals or sexual traits that are shaped by sexual selection. And by sexual selection here, I just mean exactly what I've been talking about in the past few slides, where you can use these traits to choose a particular mate in a species. So it's species specific coloration. So from this, the biology of color is really diverse, and it's not really easy to study because color patterns are tuned by variety of selective forces. Basically, you have to tune colors so that you can maybe keep predators away, but you also have to tune the colors so that your same species individual can still recognize you as the same species, right? So we see that anti-predator differences and thermoregulatory functions, which I didn't really get into in this talk, are under natural or ecological selection. And UV reflective fluorescent iridescent patches, which I spoke about in the later end, was basically used in mate choice and shaped by sexual selection. So there's a sort of balance that is coming up with natural and sexual selection, both acting to shape the color patterns that we see in butterflies, and not just butterflies, in most organisms actually. So the next question that actually then we can go to is trying to understand how these selective regimes work. What really makes a patch maybe predator specific or conspecific specific? Like how do we really decide what is how these selective forces act? But before we go into how these selective regimes can function to shape the diversity that we see, I want to take a few again, a sidetrack into a different area where we talk about how we really measure coloration. So it's fairly easy to describe something as red, green, and blue, but that's not the limit of color that we see. And as humans, where we describe color with a very subjective sense, that we identify prominent colors as what they might be, but the gradation that we see or the spectrum of colors may not be exactly how each one of us would describe it. What I mean by that is that we have, for example, this circle on the left-hand side, which most of you all would say is blue. And if you take this circle on the right-hand side, I'm sure most of you all will say that it's green. But what about this color, right? Is it bluish green? Is it greenish blue? And depending on who you ask, you can get different kinds of answers. And some will even say it's a separate color on its own. So the idea here that I'm trying to sort of get to is that colors can be confusing because depending on who you ask, you can get different answers because it's a very subjective way, subjective sense for humans. So how do we really go into classifying colors objectively, right? So how do we study color in any system in an objective way, which is the same for every person who sits and measures these colors? So there are two ways in which we can go about quantifying color. One is through digital photography, and the other is through a method that we use in the lab, in our lab, called reflectance spectrophotometry. And I'll talk in brief about digital photography and go more into detail about reflectance spectrophotometry, because that's what we use in our lab, where we have in digital photography, we use a digital camera and we have a standard. So basically a standard is nothing but will remain the same under any environmental conditions, because when people take photographs, it's highly dependent on the environmental lighting that is there, and under different kinds of light you can get different kinds of images. So you need to have a standard that can tell you under all lighting this gray is going to remain gray. So you take an image with a digital camera and a gray standard, and then you have a number of software, which I'm not going to be getting into, but a number of software which you can process these images with. So essentially what you use to establish color here is a photograph. But in reflectance spectrophotometry, we use a spectrophotometer, which is an instrument that sort of generates light and can also collect reflected light, and basically whatever light is reflected from the surface of your object of interest. And then what we get is a reflectance spectrum. I'll get to the reflectance spectrum in the next slide, but it's a spectrum that indicates what wavelengths of light are being maximally reflected from your surface of interest. And again, these are processed with different kinds of spectral software on a computer. So let's get into what exactly reflectance spectrophotometer is very briefly. So this is the reflectance spectrophotometer that we use in our lab. This is just an image, a representative image of that, which we use to take measurements of our butterfly wings. So over here we have the light that is coming out of the reflectance spectrophotometer, and it falls onto your butterfly wing, and whatever light that is reflected from the wing is collected by a detector, again by the spectrophotometer. And then this spectrophotometer will send whatever that reflected light is to your computer with the software, which can process what the information is in terms of reflectance. And after I do some tweaking and modifying of the spectra and try to clean up the data that we get, we finally are ended up with a reflectance spectrum here, which I'm illustrating. And if you look at this diagram, on the y-axis here, we have reflectance, which goes from 0 to 100. And basically 0 means no light is being reflected, that it's probably like a super black object. And at 100, we see that all the light is being reflected. And on the y-axis, on the x-axis, we have wavelength. And normally what humans can perceive is from 400 to 700 nanometers. That is the violet to red range. But this spectrophotometer is also capable of producing light in the UV range up to 300 nanometers. And we can also collect that data here. So, and if you notice here, this is the spectrum of what is a black color on a wing. There is not much light that is being reflected because it's a black patch. So this is how we go about collecting data in the lab. So with that idea, I'd like to just take the next few minutes to go through some of the work that we have done in the lab to explain how selective regimes can shape the function of traits, and how the understanding the functions can actually help us understand why so much diversity in color patterns has come up. So we use a group of butterflies in the lab called the papilio swallowtail butterflies, and to study the evolution of color patterns. And these butterflies are a very interesting group to study, because they have mimicry that is widespread and sexual dimorphism as well that is present in these butterflies. And if you remember from the previous slides, mimicry is defensive. It is defense related, and sexual dimorphism is usually sexually selected or used in intraspecific communication. So here I'm just trying to illustrate a few of the species of papilio butterflies that we see, where we have non-mimics, basically no mimicry in these butterflies in these 3 rows here. And over here we see a species that I actually showed in the previous slides, where we have one non-mimetic form here of males and females, and a mimetic form here, which is present in this species, along with one of the aposomatic models that we had spoken about again earlier, that exhibit that are toxic and birds and other predators generally avoid. So, and another interesting thing apart from mimicry and sexual dimorphism is that we observe that under different kinds of illumination, these white patches actually appear slightly different. So if you look at at, at the visible column here, which I've tried to highlight, we see that all of these patches appear not white to us, right? But when we look at it under UV illumination, some of these patches are fluorescent and some of these patches are UV reflective. Again, these are kinds of patches that I spoke to you about that butterflies have. So we see that some of them have fluorescent patches, some of them have UV reflective patches, and this is true even for the models that we've got here. So what exactly is happening here, right? Why do we have the different kinds of patches which appear white under visible light, but under UV illumination have two different kinds of properties? So going into that, the first thing that we tried to do in the lab was try to characterize these white patches in these butterflies. And how we did that was we used reflectance spectrophotometry. And basically we shined light on each of these white patches in these different butterflies, and we collected whatever reflectance that we got. And here in panels B, C, and D is is the data that we have received or collected from these different measurements that we've taken. And I'll just go through this diagram a little bit in detail because a lot of interesting things come up here. First is that we measured both the UV reflective and the fluorescent patches, and we see that these fluorescent patches have a different reflectance and the UV reflective patches have a different reflectance. Here indicated by the purple lines are the UV reflective patches and the green lines are the fluorescent patches. So clearly from this itself, you can see that these patches have some differences to them. On top of that, we also noticed that these green lines are mainly associated with these non-mimetic individuals or non-mimetic species. And these purple lines are present in the mimetic individuals as well as the models, which have a similar kind of UV reflectance in their white patches. Furthermore, just as an interesting point, if you notice that this is actually a panel of the female butterflies, and this is a panel for the male butterflies. And we see that there are more number of mimics indicated by the purple line here in females than in males. Again, going back to the dimorphism argument between males and females that I had illustrated in the previous slides. And finally, just to compare with their aposematic model here, we see that even the models have these UV reflective patches and they're not fluorescent. So now we have these two kinds of patches. The next thing to really understand is what are the functions of these patches. And that's what we tried to establish through a few experiments that we did in the lab again, where we established the function for the fluorescent patch through a mate choice experiment. So as I mentioned, these patches can be used as indicators of quality of the male, where females can use these patches to select individuals to mate with. And that's exactly what we did. We carried out a mate choice experiment where we had butterflies flying around in a cage, and we recorded their how many matings basically each individual received. We did this for one species of butterfly. It's Papilio or the common lime butterfly, again very common in Bangalore. And we basically, to give you all an idea of how this experiment worked, we had males and we split them into 3 different groups here denoted by 1, 2, and 3, where we did not modify the white patches in group 1, and we modified the white patches in group 3. And group 2 was an intermediate group where we modified the black patches just to act as a control. And we let all of these butterflies out in a cage, and we tracked them for like a month almost, trying to see how many matings each individual had received. And the idea here was that if these white patches are important to females, the males with intact white patches should get more number of matings than the males in which the white patches were modified. And that's exactly what we see when we actually plot the data. And just to go through the graph here, we see on the y-axis the number of matings, which is basically did the male receive 1 mating, 2 matings, 8 matings, how many matings did a male receive? And on the x-axis, we see treatment, which is the same 1, 2, 3 treatments here. And the ends here are the number of males that we had in each group. And from this experiment, we found out that this white color patch is important as a sexual signal, and females do use this to choose males. And hopefully from this, we can see that it is the fluorescent patch serves as a sexual signal that is shaped by sexual selection or mate choice. Now we've identified the function of the fluorescent or the one kind of view of these white patches. The next thing is to sort of understand the function of the UV reflective patch, right? We've because that was the second kind of patch that we had in these butterflies. So in order to understand what these UV patches are doing, we took the reflectance data that I'd shown in the first slide, in the first figure from the work. We took the reflectance data of mimics, of non-mimics, and their models. Basically, we have the reflectance of fluorescent UV reflective patches, and we put it in our computer and we do some simulations, and we use a visual modeling system of birds. So we know that birds have certain kinds of receptors, and they can tell the difference between UV and non-UV patches, for example. So we use that information along with the reflectance data that we collected in the lab. And we found basically that these UV reflective patches improve the mimetic resemblance of the mimics to their models. And what that really means here is that if you look at this graph, on the y-axis, again, we have similarity with the model. So basically how similar or how close is that white patch of a mimic or a non-mimic with its model. And we found that in most of these cases, actually in all of these cases, the mimics tend to have more similarity. Here, a lower value means there is more similarity, and a higher value means there is more dissimilarity. So if you look at the graph here, in each of these groups, we see that the mimics have a higher resemblance or look more like their models than the non-mimics do. And through this, we have established that the UV reflective patches make the baits and mimics look more like their aposemantic models. So from all of this, we have now identified the nature of these patches, that they have two different spectra and do have different properties, one being fluorescent and one being UV reflective. We've also identified the function of a fluorescent patch versus the function of a UV reflective patch. And hopefully now we can see that the UV reflective patch actually improves mimicry, and therefore is something that is a trait that is selected for by natural selection. So we have two contrasting selective pressures here, one for the fluorescent patch, which is sexual selection, and UV reflective patch, which is natural selection. And this sort of just goes back to tying up the story about how diversity can be explained through function of these color patches. And sort of just to summarize the findings from the studies that we have conducted, is that both natural and sexual selection have shaped the evolution of white patches in this group of butterflies, with their function changing depending on what kind of patch they are. And using reflectance measurements and behavior and modeling experiments, we have identified the functions for these white patches. And we show that the UV patch is actually now being used as an anti-predator color patch, as opposed to what is conventionally thought of as a sexually selected trait. It is actually now being used as a naturally selected trait. So we also show that that switch happens here in this butterfly group. So therefore, there is this constant interplay of sexual and natural selection that works on these butterfly wings to shape the diversity that we see across different species. And so with that, I'm actually coming to the end of my talk. And I want to end with sort of motivating this idea that color science still has a number of open questions. We have basically studied it in butterflies, and I have looked at it in a very specific group of, or a subset of butterflies, which are the papilio swallowtails. But there are so many more open questions, because an extension of studying studying coloration is that you also have to study the receiver, right? You have to understand what the receiver sees and whether a color patch is even visible to a particular receiver of interest, because we need to understand what they can see to see whether the color patch is actually important or not. So an extension of that is understanding the visual system of the receivers, which means basically trying to understand how organisms perceive color. Like, how do birds see color? How do bees see colors? How do lizards see colors? All of these different kinds of questions come up then when you're trying to understand how color evolves. And apart from these sort of large scale studies that we do, a lot of studies are directing their focus to understanding the genetics of color production in different organisms, basically trying to understand what genes are involved in producing one color versus another. And if I take the example again of the work we did with papilio swallowtail butterflies, is it the same gene that is producing the fluorescent color versus the UV color? Is it a different pathway? So all of these questions still remain unanswered in a lot of organisms. And also, of course, whatever we show in the lab, we also have to really try to validate that in the field and go out and do field experiments. And though they are tough, though field experiments are tough, they are also very important to really making sure that we have the entire picture of how color is evolving. And so with that, I hope I have left you with more information than you came in with to this talk. And I would like to thank here my advisor Krishnameg and the lab members here, and also the NCBS communication office, which is basically composed of Sonal and Harsh, who have put together this talk and been a very important component of making sure this big butterfly month goes off with interesting talks, and hopefully more interesting talks will be coming up in the next few weeks. With that, I think I am done, and I would like to take questions, if any.
Hi, Bhavya. We have a few questions. I'll read it to you.
Yeah,
Read them out
yeah.
to you.
Sure.
So this question is by Sudha.
Yeah.
Does mimicry lead to mating attempts between the male of the aposematic species and the female of the mimic species?
Yeah, so that's a great question. And the interesting thing is that I experiments with that kind of behavior, mate choice has not been done. And that's actually some of the things that I think more people should be focusing on and trying to understand how these, how does this species interactions, cross species interactions really work. But to answer, like long answer short, there are no experiments. We don't know how that really happens. So species recognition and mate choice together has not been really looked at. So
Great.
yeah.
Okay, I'll read out the second question, and it's also by Sudha. It is interesting that males of many butterfly species do not show mimicry. Does this negatively affect their survival or their recognition by females of the same species?
Negatively affected. So in most cases, the reason that people say that the males don't mimic is because they are sort of the ancestral or the original color pattern that sort of continued existing, and females had to face predation pressure because they also have to deal with laying eggs and other costly activities that males don't have to. So females tended to evolve mimicry and other such things, but the males did not. But whether it negatively affects the survival is that it doesn't necessarily negatively affect the survival, because females anyway have to mate, right? So and if there's only one kind of male that is available, females will always be mating with that morph or that particular color of the butterfly. So it doesn't really, I think it doesn't really negatively affect the survival. And recognition, again, is a very interesting question that I don't think a lot of people have looked at with respect to what exactly are the females looking at when they want to identify the same species and vice versa. So the dissecting that has not really been done to a great extent.
Okay,
Yeah.
the next question is by Sukriti. The PX lamp illuminator and the detector were situated at a 45-degree angle. Is there any particular reason for maintaining that angle?
It's a standard angle that is used, but it's not a hard and fast rule that you have to keep it at 45 degrees. You can change the angle of the detector to 30 degrees, 40 degrees, and you can still take measurements. The important thing here is that you keep that angle constant whenever you're taking a set of measurements, so that there's no variability in the reflectance at different angles. So you can do one set of experiments at 45 degrees, you can do another set of experiments at 30 degrees, 80 degrees, whatever, basically. So it's just a standard orientation that we use in the lab, but it is amenable to change.
Okay, we have two more questions.
Yeah.
So Asma asks, just like white patches, can we see UV patches naturally in butterflies or only under UV light?
Yeah, no, we humans don't have the UV receptor. We only have red, green, and blue photoreceptors in the eye. So we can't see UV except with the help of UV filters, which sort of only allow the UV to pass through. And that also has to undergo some transformation so that we see it as UV reflective. And we have to cut out all the other light to be able to detect this. Another thing is that these patches are often are not as bright as these visible patches. These UV patches are usually duller, have lesser intensity of their reflectance. So it just adds to the difficulty to see the UV patches. Plus, we can't see it without the help of filters. So yeah.
Okay, this one has been asked anonymously. How do mimics arise? Is it by chance and then selected for, or is it a more directed process?
It's mainly by chance. There is no real direction that evolution really takes. There's no like set path on which if you go do A, B, C, D, things will become a mimic. It's often a random walk around whatever color patterns that it samples. And for example, if you take one species and it sort of develops a red color and it looks closer to a toxic individual, but at the same time, there is another individual that has a mutation that makes it look slightly whitish. In that case, the red one is getting a selective advantage here because it resembles a toxic individual, a toxic species, and therefore through multiple steps of refining this color, it can eventually be a mimic in the mimicry ring. But the white one, for example, might either be completely removed from the population or it may remain in very low frequencies. So there's no like direction, and it's often an iterative process of generating multiple kinds of color patterns, which then result in a good enough resemblance. So, yeah.
Bhavya, do you have time? We have a few more questions now.
Yeah, I think. I think until 7. I mean, we have 5 more minutes. We can take a few more questions.
Okay, I'll read it down.
Yeah.
So Jigisha asks, in a peacock, male has long feathers for attracting female. Likewise, any other features of butterfly for attraction?
Yeah. So if we go back to the slide where I was talking about the UV reflective patches on butterflies, a lot of the time these UV reflective patches do function as a indicator of like attractiveness. So not just that, there are also visible patches, and if you do, a lot of experiments have been carried out where they actually block out some color patches or they remove some scales and make the patch less bright, and then they see that males with less bright patches or less or where similar to what we did with the swallowtail butterflies, if you remove or modify these patches, those males tend to get lesser matings. So that basically is an indirect assumption, indirect conclusion that males with intact patches are more attractive. But again, it could be UV reflective, it could be iridescent patches, and it could be these fluorescent patches. But there's no like one set patch that does that.
Okay.
Yeah.
Next question is by Ayush. Were the UV patch manipulated in the first experiment, confirming that fluorescent patches are sexually selected?
No. So the experiment that we did to modify the fluorescent patches is only done in one species of butterflies until now, and that species only has these fluorescent patches. It doesn't have the UV reflective patches. So modifying these patches only was done with the fluorescent patches and not the UV patches. The UV patches we used to do the visual modeling of predators, and then we saw that the mimics had better resemblance with the models than the non-mimics had. So that's the idea, that these fluorescent patches might be having a different function and the UV patches have a different function.
Okay, we have another anonymous question. Does the amount of glossiness of the wing impact sexual selection?
Yeah, I think I answered that in the previous question. Like it definitely could, if you are less bright, if you're less glossy. All of these parameters could definitely work into trying to into more affecting mate choice in these butterflies. So yeah.
Haley, she has a question. Do environmental cues regulate coloration? If so, how does it connect to the question of function? As in, does coloration change when location, habitat of a species change?
Yeah, so environment does regulate coloration, but in a different sense that I haven't really gotten into in this talk. where depending on the temperature and humidity, sometimes color patterns change. To take the example of a very common butterfly that we see here, which is the evening brown or Melanitis leda, we see that under dry season or when there is less rain, it has a different wing pattern versus under wet season when there's a lot of rain and probably temperatures are cooler, it has a different wing pattern. But that is a different strategy that I have not spoken about, where environment can regulate the coloration that we see in individuals. And when another way in which it can regulate color is when, as I briefly mentioned, the idea of thermoregulation. We see butterflies at different, say, altitudes. So the same species at different altitudes can have different kinds of color patches, because in the lower altitude, they might be less black to make sure that they're not overheating. But in higher altitudes, where the temperature is lower, you could have more blackened individuals or more melanistic individuals, which can actually more effectively trap heat and therefore help in regulate the temperature that way. So yeah, environment also does have a lot of influence on color patterns. Along with environment, even sometimes food sources with where when caterpillars are feeding, that could also affect the quality of food, could also sometimes affect the quality or the amount of wing pigments that have been deposited in adults. So that's just a side point.
Okay. Probably we can you can answer last two questions and then we can close the session.
Yeah,
So
sure.
Preeti asks, is there any geographic variation in mimic patterns by specific species? Because different places have different predator bases and their color perception mechanisms may differ as well.
So geographic variation in mimic patterns by a specific species. So the thing about mimicry rings is that they are isolated. They depend on the model and the mimic being present in the same geographic location. So you can't, most of the times you won't find a mimic that is present outside of the range where the model is not present. So for example, if you have a model that is distributed only in like the southern part of India, often you may not see the mimic present in the northern parts without the model counterpart. And as per the different predator bases and their color perception mechanisms, so broadly speaking, it's mainly birds that we consider as predators of butterflies. And studies have shown that there are two main kinds of visual systems birds have. Some subset of birds can see UV blue, red, and green. Some birds can see a slightly shorter blue, that is like they have violet, they have blue, red, and green. And this is fairly conserved across a lot of bird species. So ideally, there should not be too much difference between birds in different geographical regions, because both kinds of birds will exist in all of these areas. But if you consider other kind of predators which are non-avian predators, then we're not really sure what's happening there, because we haven't really studied predation of butterflies in context of non-bird predators. But that could change, and that would be another interesting question to see if say mantis or lizards can that feed on butterflies also care about mimicry in that sense. But mainly it's bird related, and it's fairly conserved, the visual systems of birds. So
yeah, and I think we can take the last question. So
yeah.
If this is a Jigisha, if I catch the butterfly, I get a print on my finger. Why do I get that print?
So actually, those are the scales of the butterfly that are coming off on your hand, and that's why, like in the lab, we don't really catch them with our hands, because a lot of the each of the scales will come off. So that's nothing but these structures on the wings that these pigments and other basically pigments are deposited on, or the scales that are arranged in certain ways to give you iridescent coloration. But yeah, those are nothing but the butterfly wing scales, and we don't hold them with our fingers in the lab. So to prevent that from happening. But yeah, I think I think that's all, right?
We do have a couple of questions in the chat, but like it's up to you. If you can you see them? One from Jyoti, why the small butterflies flutter more? What's the reason behind it?
Sorry, could you repeat that?
Yeah, Jyoti asks, why do the small butterflies flutter more? What is the reason behind it?
It's probably because they need to do more work to probably traverse the same distance as a larger butterfly. So, in from moving from point A to point B, a larger butterfly would probably have to flap its wings maybe twice, but a smaller butterfly, because of its small size, would have to flap multiple more times to reach across in that same distance.
Okay, and the one final question is I was just wondering that a bird sees butterflies differently with the human,
Yeah.
and mimicry helps butterflies to hide. Did you compare the difference between these pictures?
Uh, I don't I don't understand that question really. Difference between pictures in the sense of through bird vision and human vision, because if that's the case, we did that with the papilio butterflies, where we did take the bird vision into account, and we modeled how the bird sees these patches. So yeah, in some sense we did compare the difference in human and bird vision in that sense. So
Great, I
but
think.
yeah.
Yeah. Sorry, you can
No,
continue.
that's it, I'm done, thanks.
Okay, so we can, I think, close the session. I have shared a feedback form with everyone. Please do
Sure.
fill it.
Yes.
And thank you, Bhavya, so much for the great session. I think the
Thank you for
attend
attending
yeah,
and
the
organizing
attendees
it.
enjoyed it a lot, as they're saying in the chat. So,
I'm
okay,
glad. I hope they learned more things than they came in with.
okay then, everyone have a great day. I'm closing the session now. Bye,
Thank
Bhavya.
you.